Good afternoon viewers and welcome to the EduSat live lecture for the science stream. Today we are going to talk about a very important hormone, ethylene. It may sound to you as an organic compound, but this is another plant hormone which we are going to study, and we have here with us Dr. Ashish Kumar Nandi from School of Life Sciences, Jawaharlal Nehru University. So Dr. Nandi will be talking to us about what are the roles of ethylene, what is the mechanism of its action, and what all processes are regulated by this important hormone. So continuing this series on phytohormones, we welcome Dr. Ashish Nandi.
Thank you, Dr. Unnati. So this is a continuation of the series of plant hormones that I have talking in this channel for last quite a few lectures. I think maybe 6th or 7th lecture. And this is about plant hormone, as Dr. Unnati said, ethylene, and I am quite sure most of you have heard about ethylene as a plant hormone, because it is one of the classical plant hormone. This hormone has many role in plant development, like senescence, fruit ripening, seedling development, root development, and in addition it has a special duty in controlling plant disease defense. So this is also considered one of the key plant disease defense hormone. So today I will speak about its discovery, its receptors, its signalings, and also how it was discovered, which I normally speak of for any hormone. So let me begin with that, and also again I am reminding to all of you, like if you have any question, any query, study material, please do not hesitate to write me. I am showing my email address almost in every class. Today also I will show you. Okay. So now let me begin with ethylene, and knowingly or unknowingly the ethylene hormone is being used in fruit ripening for ages. It has been reported since say 1850 onwards that people try to enhance fruit ripening or delay fruit ripening. One of the reason is that the difference between the farmer's land to the market. So the fruit must be harvested when it's hard and brought to the market so that they do not get damaged or destroyed on the road. But when it reaches the market, it should be ripe so that it is palatable, it is attractive to the buyers. And it has been reported that very early ages people used to keep fruits and some kind of incense sticks. That incense sticks would burn, make some kind of gas, and it seems it enhances fruit ripening. And then even in early days people used to make wounding. There are reports that if a lot of fruit is kept in a room and some of the fruits are made deep cut and wounding, those wounded fruits would release ethylene, and that would enhance ripening of the other fruits. So those are the time when people did not know that it is ethylene. And even today, like most of the fruit sellers, vendors in many cities like Delhi, or even the farmers, they used some chemical called ripening chemical. This ripening chemical is nothing but some kind of carbides which would react with water and generates ethylene gas inside the tissue, and that would enhance ripening. So this is something, a common man's hormone, which a lot of people use together. So let me talk about today how it was discovered and how it really works. So this is, I am showing you my email ID. So any of you, if you want to any kind of study material, please do not hesitate to write me. So this is my email ID, which is written here, ashis_nandi@yahoo.com. So this is the hormone list which I have shown you many times, and today I am talking about this ethylene. So the upper 5 ethylenes I have, this is the last one I have talked. I have finished auxin, cytokinin, gibberellin, and abscisic acid. So, this is the ethylene, which is also a classical hormone because it is known from ages. So, this ethylene, as this slide shows, that it has a role in senescence, leaf shedding, ripening, seedling development, organ expansion and stress responses. So, now look at this beautiful picture. You will see that a leaf, a plant undergoing senescence here, it looks so colorful. Actually, this is a process by which plant take its resources. So, a green leaf is full of energy, full of metabolites, which many of it can be used. So, during senescence, plant break down those products and take it and store it mostly inside the stem when entire leaves falls, or sometimes in the newer leaves. And what is retained here are the cheap chemicals, like most of them are hydrocarbons and anthocyanin pigments, which are quite cheap for the plants, and they leave it there. So, they look very beautiful, and lot of people go to see this beauty of nature, but it is a natural process for reusing the energy. A very similar thing happens during fruit ripening, where fruit initially remains green and then eventually ripe, and during fruit ripening a fruit undergo many changes. As I told earlier, a hard fruit must be soft. So, there is a lot of metabolic energy is required, and the fruits, most of them are made of cells like parenchyma cells. These parenchyma cells are attached with each other by middle lamella having pectins. So, the ripening fruit secretes this enzyme like pectinase that would remove this hardness from the fruit, and so there is a lot of respiration requirement, a lot of energy requirement, and those are the things which are provided by hormone like ethylene. Ethylene enhances this metabolism and this respiration rate. So, this is what is role by ethylene. Now, this is a beautiful observation by a scientist in 1910. What he found that when he sipped orange and banana together, he found banana ripened much faster than when it was not with oranges. Of course, later on it was found that in his orange stock there was one orange or some oranges which had the fungal contamination. So, that actually ripening oranges is going to release ethylene anyway, but when it is fungal infection is there, then it is even more. So, this fungal infected oranges released much more ethylene, which enhanced ripening of banana, which was shipped together. But anyway, so it was not known, that time it was ethylene. So, this is something quite interesting. One Russian PhD student named Dmitry Nelezovo, he made one wonderful discovery. So, in that period of time nearing 1900, the home or street lights were mostly had a gas lamp. So, here actually I am just showing one photograph, but this is not the person, I do not know, but I am showing, just look at this gas lamp which were used inside the home and as well as street light. And this PhD student Dmitry was studying growth of pea seedlings, and what he found, he was looking the light versus dark, and in this experiment he found his pea seedlings behave differently when it was kept outside the home versus when it was kept inside the home. So, I will show you in the next slide. So, this is what normally happens. This is not from this Dimitri's paper though. Like here, a seed just germinated in presence of light, and this is what is called as photomorphogenesis. Now, if the same seedling is kept in the dark, then one thing you would see, there is a much elongated hypocotyle. This is hypocotyle elongation. Now, if this seedling is kept with increasing concentration of ethylene, they will say that there is a much decreasing in the ethylene content, and thickening of the hypocotyl. So, I will come back to this phenotype later, but this is what something was observed by Dimitri. So, Dimitri was very intelligent. Then he must thought that something may be there in this burning lamps, because the difference between outside and inside is the burning lamps. So, he analyzed the gases which come out from the burning lamp, and he found it is rich in ethylene. So, he concluded that it must be ethylene from these burners which is making the pea seedling behave differently, and he was absolutely right. Even he observed that that time actually the streets used to have the gas lights, and he observed that many trees which grew aside of the gas lamp had the leaf shading much more than the plants which grew away. So, he concluded that definitely something is there, maybe ethylene, which is promoting a leaf shading and as well as the pea seedling defect. Much later, around 30 years later, a scientist again found that ethylene is also present in ripening apple. So, this is a Nature paper. First time he showed ethylene is not only present in the gas burner, but when the fruit ripen, this ripened fruit also make ethylene, and that is what is an endogenous hormone which promote fruit ripening. This is a paper from Plant Physiology. I am just showing this paper because you can appreciate how people meticulously identifies hormones. Look at the diagram from this paper, and this is I am not going to explain in details. Some of you, if you want, I can send you the paper, but see, this is the avocados. There are large number of avocados were kept in a barrel and allowed to ripen. So, it will make out ethylene, and then he passed to many columns, many absorbents, and then finally took ethylene, and then ethylene he was analyzed by chemical reactions, that how much ethylene is made by this avocado. And that way he also confirmed that it is not nothing to do with apple, many other fruits including avocado also makes ethylene. Much later again 1959, 1960, the gas chromatography came up. This gas chromatography is much millionfold more sensitive. So, this is a gas chromatography, you can see the spikes. So, this is for ethylene, this is for air, and a very little amount of ethylene can be detected by gas chromatography, and then lot of paper came which showed how ethylene is so important and how much ethylene is made. Here this paper in Plant Physiology also made lot of important discovery. One of them is temperature. What has been shown here, if you look at this graph, so here is one line which showed amount of ethylene when it was kept at 20 degree, and this one you can see the amount of ethylene at different temperature, starting from 5 degree to 35 degree. What this scientist showed that as the temperature increases from 20 to 25 to 30, amount of ethylene also increases much more, and that also correlates with the speed of ripening. So, speed of ripening is enhanced by external condition like temperature, and it is also coupled with higher amount of ethylene production. This is another paper in 1962. This is also a good paper which showed very important thing. As I told you earlier, the process of fruit ripening requires a lot of energy, so the respiration rate has to increase. When the respiration rate increases, then carbon dioxide amount is going to increase. So, this paper also used GC, gas chromatography, and measured amount of carbon dioxide versus amount of ethylene. Now, look at, and this is the time scale in the x axis. The most important point to take from here is carbon dioxide peak comes later and ethylene peaks come earlier. That means the ethylene spike or ethylene pickup is important before the respiration is pick up. In other words, this is something which happens before the actual ripening process takes place. So, that is what I have given the title here. GC revealed that ethylene is a cause, not a consequence of ripening. That means ethylene comes before actual the ripening process begins. And this is a 1979 paper, which is also very interesting paper that elaborated the ethylene biosynthesis mechanism. So, in here actually by that time many papers came out, and scientists have shown that you can provide 14 carbon methionine, and this 14 carbon methionine's carbon actually can convert into ethylene. So, you can actually emit radioactive ethylene, you can measure radioactive ethylene, and you can study and trace how carbon flows from methionine to ethylene. And here in this graph you can see the amount of ethylene produced in a ripening apple slices when you keep in nitrogen. Look here, the lower one is in nitrogen. That means when you keep in nitrogen, inside the nitrogen only, then there is no ethylene production, ripening is much delayed. If you keep it in air, then ethylene production picks up very early, ethylene production rapidly increases over time. And look at this graph, very interesting graph. It was kept in the nitrogen until this arrowhead, which is around 9th day, 8th day. What they have noticed that until 8th day or until it was shifted to nitrogen, there was almost no ethylene. But the moment it was transferred to air, then ethylene started picking up high amount. So, the conclusion is you need air or most likely oxygen for ethylene production. And so, from the previous study one can conclude that you have methionine. This methionine must be converting to some compound which was not known. So, in the paper they wrote as compound X. This compound X most likely reacts with oxygen and provide ethylene. So, this is what a big discovery at that time in 1979. And look at this paper which came, this is also same paper in 1979. Then what they did is, this one I explained earlier. So, this is the time when nitrogen to air is picking up. Now, if you take sample from here, what will happen? The compound X will be accumulated. See, methionine converting to compound X converting to ethylene. So, if you keep under nitrogen only, it will accumulate compound X, but not ethylene. So, what they did by GC, they have taken the one which was in air, one in nitrogen and nitrogen to air, and look at this peak in the GC spectra. This peak was present only when it was kept in nitrogen. The moment it was transferred to air, this peak is vanished, reduced. So, this is the peak which was compound X, quite natural conclusion, and the identified compound X in this paper turned out to be something called amino cyclopropane carboxylic acid or ACC. So, this was a big discovery in terms of biosynthesis of ethylene, and quite interestingly, from many chemical companies today including the Sigma Aldrich, you can buy ACC which is very cheap, and you instead of application of ethylene you can just apply ACC. The one of the reason actually why lot of people use ACC in the experiment is ethylene is gaseous hormone. So, when you have to apply a gaseous hormone, you must keep the plant under cover, otherwise ethylene will evaporate away. However, ACC is a solid in room temperature, it is a salt kind of thing. You can dissolve in water and apply to the plant or on the medium, and then this ACC will convert into ethylene inside the plant, and that will promote the ethylene responses. So, this is in brief how ethylene was discovered, ethylene biosynthesis pathway was discovered. Later on, actually, this is two scientists, one is Tony Blaker, one is Hans Kende. This Tony and Hans, they contributed a lot, mostly by using genetics in Arabidopsis to identify the genes and the enzymes which makes this process. So, this is methionine. It forms one intermediate called adenosine-methionine, S-adenosine-methionine, also known as SAM quite often, and the gene which makes ACC is called ACC synthetase. So, this is called ACS, and then ACC oxidase ACO, which converts into ethylene. One thing also you can observe here, this oxygen is required which converts into carbon dioxide in this process of ethylene biosynthesis. So, oxygen is produced for ethylene production, carbon dioxide is also produced in the ethylene production, oxygen is consumed, and also carbon dioxide is produced due to respiration later on. And a brief about regulation of ethylene gas biosynthesis. See, normally every time when I am speaking about hormone, I am also speaking about their regulation and conjugation, degradation and biosynthesis, so that what you call as a homeostasis. So, this paper came in 2004 in Nature. They identified a very good regulator which they called as ETO1. The name of the gene came as ethylene overproducing 1, and to most of you who may not know that in plant the gene name is quite often given by the mutation name. So, if you have a mutation in this protein ETO1, then you get a lot of ethylene. So, this is an ETO mutant phenotype. Maybe I will speak to you later on about this seedling phenotype, but what you can see here that this wild type, and if you have a ETO1 mutant, then there is a lot of ethylene inside the plant. So, it behaves like as if the plant has been treated with ethylene. So, it makes a lot of ethylene, and this is the one of the slide which I am showing you just to show how regulation takes place. This is bit complex, but let me briefly tell you. This is the ACS, is a protein, codes for an enzyme. This ACS dimerizes and make a functional protein enzyme for making ethylene. This one, when it binds with ETO, there is a ETO, when it binds, then ETO does not allow it to function. So, along with ETO, this is inactive, it does not make it, and when signal comes, ETO breaks down. When ethylene production signal comes, ETO breaks down, ACC is free, that converts SAM to ACC. That is what the role of ETO is. So, it is a negative regulator which regulates and makes sure that a plant do not make ethylene unnecessarily. It will be made only when it is required. So, this is ethylene degradation step. Ethylene is very vulnerable for oxidation. So, ethylene can have many oxidized steps. Ethylene can convert into ethylene oxide, ethylene glycol, oxalic acid, and eventually to carbon dioxide. So, this is also quite interesting because this is if you somehow can provide oxidizing agent, like one of the common oxidizing agent used in the laboratory are potassium permanganate, KMnO4. If you use KMnO4 for example, which can oxidize ethylene, then you can break down ethylene. So, this is another way to remove ethylene. So, it can also have the applications in case of delaying ripening. So, apply KMNO4, ethylene will be degraded and that will not allow to ripen immediately. So, this is a way one can manipulate even outside the amount of ethylene amount. So, once the ethylene was identified as the fruit ripening agent, and one can also buy it not a very expensive price, lot of people have tried to control fruit ripening. This is a cartoon here. You can see this lady is using an ethylene cylinder here, and this is a chamber. Inside the chamber she kept many fruits and trying to ripen the fruit by using the cylinder. Today, if you see, there are many sophisticated setups are there for controlling fruit ripening. Like from the farms, the fruit like banana will come as a truckload and will reach to the city. In the city, they have something called ripening center. In the ripening center, they have ethylene cylinders. This ethylene cylinder will pump ethylene, and at the same time it also will take away carbon dioxide or monitor the carbon dioxide concentration, because they do not want too fast ripening as well. So, ripening must be a slow process, but it should be also enhanced by ethylene, so that you get a very nicely color developed and inside it is palatable, starch degrades, breaks down to glucose. So, the fruit is sweet and this is done. But however, this is quite expensive, so most of the marginal farmers cannot afford this one, and its fruit ripening is now a technology. You see, there are some setups where you have a supercomputer. So, this is a computer, and this computer controls how much of cooling, because temperature is important, how much of heating and how much of ventilation, and ethylene, carbon dioxide, oxygen, everything is controlled by computer, so that ripening process is perfectly made before banana reaches to the supermarket or any other fruit. So, this is a cartoon just to show you that even scientists are trying to get that laboratory inside the trucks. So, like a truck carrying green banana from the farm, and by the time it reaches the market, it should get automatically ripened. So, this is what it looks like, that scientists are trying to control oxygen, carbon dioxide, ethylene and everything, which should be on a rolling truck, so that one can control in a way that before reaching to the city, it is just ripened. Fruit ripening and ethylene and senescence also is normally shown in many laboratory and as a students' practical classes. Like here, you can see a beaker is covered on inside the flask, and flask has the twigs of the leaves, and another beaker covers similar one where there is an apple which is allowed to ripen there inside. Now, in this case you can see this ripening apple will release ethylene, and that will allow senescence, and that all the leaves are fallen, but not in this case. So, this is also quite often even some of the teachers can do it, like a small demonstration of ethylene production in their laboratory for the students, and I think the students will really find very interesting to see that. Ethylene also has like fruits also have the applications in cut flower. See, if you have a cut flower, after few days it is going to dry up, senesce, and it can be delayed by removing ethylene. Like I will tell you in the class today, there is a receptor called ETR1. This receptor binds ethylene via a copper molecule, and silver can compete with the copper. So, if you have silver particles inside the water, the silver will inhibit with the copper, and then ethylene response goes down. So, this is STS, means silver thiosulfate. This silver thiosulfate added flowers remains viable for much longer period of time than the one which is controlled. So, this is the summary so far, is what I told you, that ethylene promotes senescence ethylene promotes fruit ripening. Ripening fruit produces ethylene, which further promotes respiration. Ethylene was identified as an agent in burning tar or oil that affects seedling growth. Ethylene biosynthesis takes place from S-adenosyl-methionine to aminocyclocarboxypropane and then ethylene. Ethylene oxidizes to carbon dioxide, and manipulation of ethylene content is used in both the ways for delaying ripening as well as sustaining of ripening. Thank you very much for your attention. I will come back very soon and tell you about the remaining part of the ethylene signaling. Thank you. Welcome back. I am continuing with this ethylene signaling. So far I have told about how ethylene was identified in a ripening fruit, and how ethylene by a very brilliant scientist Dimitri, who identified ethylene from a burning gas lamp, and which promoted and modified the seedling phenotype. But I have not spoken about how this ethylene affect seedling phenotype, and now I will speak to you, which the reason I am telling later, because that is the way how people really identified its signaling pathways, signaling components, because that is a very remarkable phenotype which one can get it and can use in a genetic screen. So let me show you these phenotypes, what happens when you apply ethylene. So then I think you can follow better. So here I will show you some of the ethylene phenotypes, and that is in a model plant Arabidopsis. This is wild type Arabidopsis plant which has no modification, growing normally. Here is a small plant where ethylene production is enhanced, ethylene signaling is enhanced, not production. Here you can see a plant where ethylene production is enhanced. So there is a much more leaf senescence than wild type plant, and similarly the plant in the right has more ethylene. So there is a short root and many root hairs. It is not only at the visible phenotype, you can also see many expression of many gene switcher changes. So here two genes which are shown, one is ERF1, that is a transcription factor which is induced by ethylene. So as the time passes, this transcription factor keep on increasing, and this is one of the downstream target of transcription factor. Amount of transcription factor also increases as the time passes after ethylene application. So, there are many effects of ethylene. Now, look at carefully about this ethylene effect on seedling growth. This is commonly called as triple response of ethylene. Earlier I told you that if you germinate a seedling in presence of dark, then there is elongation of the hypocotyl. The same thing if you had grown in the light, you would have maybe half of it, but in the dark it actually elongates much faster. This seedling which is grown in the dark, if you apply ethylene, then what you will find that this become very short, and there I will show you later, but you can see the tip is something different. Tip is not like this, right. So, this is called apical hook formation, and also root length is also quite short if you look here. So, there are three responses. One is called thickening of hypocotyl, shortening of root, and apical hook formation. So, this should be thickening and shortening both of hypocotyl, shortening of root and apical hook formation. So, this is what is called as triple response of ethylene. But remember, for observing this ethylene response, it is better to keep in the dark where symptoms are exaggerated. This is a photograph which is shown in a close up view. This is what happens when seedling germinates. Normally if you keep in the dark, then the tip bends. Had it been in the light, this would have been upwards, and these two leaves, cotyledonous leaves, would have been opened up. But this does not happen because of the dark. If you keep under ethylene, then this is what happens. So, there is another hook formation, it goes up. So, this is what is called apical hook formation. Now, the exact biological reason is not known why or these things happen, why there is a hook formation by ethylene. However, there is a ethylene is also known to promote gravitropism. So, somehow probably extra ethylene disturbs this gravitropism, which is normally happens without ethylene. However, the purpose as I said, though not exactly known, people somehow have given some kind of belief. This is one of the idea what people have floated is, suppose a seedling is growing under a kind of a rock which is giving this dark, and because the seed germinated just under the soil or just above the soil, maybe under the rock, this place will accumulate ethylene because there is a heating up on the rock. So, there is a kind of a wounding or stress that would cause ethylene, and this ethylene will help them to bend and grow forward this way. So, ethylene actually helps in changing the growth direction so that it can bypass this rock over here. So, that is what is not proven, but it is a proposed theory of bending of the apical tip. Now, what experimentally people have shown how ethylene really helps in shortening the growth. This is a very interesting paper came in Tony Blaker's lab in 2004. This is A.B. Blaker's. Look here, this is the diagram of the seedling tip, and this arrow shows the growth rate. This growth rate, the first, and this photograph was taken at every 30 minutes and then kept next to next. Now, you see the first 3 pictures were taken without ethylene. Look at the yellow line, the growth rate, and then ethylene added. When ethylene added, look, the growth became slow, and then ethylene removed, and again growth became fast. So, ethylene inhibits hypocotyl elongation. It is very clearly demonstrated in this experiment. And this phenotype is also used to identify ethylene mutant. like what you can see here, large number of mutant EMS treated mutant seedlings are kept on the dark and ethylene, and everything, of course everything except one is showing the triple response. Now, if a plant or a mutant one which cannot sense ethylene or cannot perceive ethylene, then it will not show the triple response. So, it will show as if it is growing in the light and without ethylene growing in the air. So, this is what is a very useful mechanism to identify components of ethylene biosynthesis, ethylene signaling, and these mutants was referred as ethylene insensitive mutant or ion mutants. Lot of mutants have been identified that way. Here I am giving you the list of them, some of them. They are ethylene insensitive mutant, and they are ETR. ETR stands for ethylene receptor. There are ETR1, ETR2, then IN4, IN2, IN3, 5 and 6, where ethylene do not impact on the growth, seedling is tall. In contrast, you can see there is a different mutant which is called constitutive triple response. This plant, this mutant will grow in air, but it will look like as if it is growing in the ethylene. So, that is what is the term constitutive is. Constitutive means spontaneous always. So, this mutant activate ethylene signaling when actually ethylene was not applied. So, I will speak to you today how why it is showing like this in a minute. This is in brief how ethylene signaling takes place. So, the receptor is ETR1, and then like many of you probably know how receptor signaling takes place. So, usually receptors are followed by a kinase. So, there is a kinase called CTR1, and there is a regulator called IN2, IN3 and ERF1, they are transcription factors, and these are the target genes. There are many target genes, many transcription factors, but I am showing only a few of them. So, this is the pathway of ethylene signaling. It is received by ETR1, then it goes to CTR1. But see here, there are at least two phases. This is a negative regulation. So, ethylene receptor itself is a negative regulator of ethylene signaling. So, that is what actually in brief about how ethylene signaling takes place in plant. Now, I will show you how this signaling pathway takes place. This ETR1 actually is a receptor which is a membrane bound. Now, you can see this is the membrane bound receptor. So, it has a transmembrane domain, and this ethylene binds in the membrane, near the membrane, and the next one is CTR1. This is very interesting. The CTR1 is a kinase, it is a RAF like kinase of mammalian system, and this kinase can phosphorylate other molecules by using ATP. So, it converts ATP to ADP and phosphorylate its targets. Now, the interesting part is that this kinase has a auto inhibitory domain. Look here. So, this is what happens when this auto inhibitory domain folds back and inhibits the activity. So, the ETR1 is the receptor that interacts with the inhibitor domain. So, I will just go through little slow. This is a bit complicated to understand at the beginning, but hopefully you will understand. If the receptors are not there, then this inhibitory domain will fold back like this. So, here inhibitory domain is folded back and this cannot phosphorylate. Now, what I told you, this is an enzyme where when it is not phosphorylating, it is active. This is the quite interesting thing about ethylene signaling. So, under normal condition, this is wild type plant, under normal condition there is no ethylene, then this ethylene receptor ETR1 will bind with the inhibitor domain of the CTR1. So, it cannot fold back. When it is not folding back, it is active, this can phosphorylate, and this phosphorylation is required to suppress ethylene signaling. That is very interesting. This is in contrast to many other kinase signaling that you have read. I have spoken about kinase signaling in cytokinin. I will speak to you today also, remind you about that, but this is in contrast to it. So, when it is phosphorylating, it is active kinase, then signaling is turned off. And this is a mutant. Suppose you have a mutant of ETR1, then ETR1 is not there, then it falls back. And many times, many of the ETR1 mutants can actually spontaneously affect this kind of signaling. Now, what happens is that under wild type condition, when ethylene binds, so this is the condition where ethylene bound, there is structural change. So, this structural conformation change relieves the inhibitory domain, and when it is relieved, it falls back. So, then ethylene signaling is turned on. So, the suppression mediated by phosphorylation is removed when ethylene binds. You can see here the Cu written, this is copper. So, as I told you that copper is an integral part of the ethylene receptor, where probably with the help of this copper ion, ethylene binds with the receptor, and that is where the silver comes into play. When silver is there, then it inhibits this copper mediated interaction. So, this is in brief about how ethylene reception takes place and how it suppresses the following kinase, which is CTR1. Now, what happens actually if you have a mutation in CTR1 which cannot phosphorylate, then ethylene signal is constitutive. So, that is why the mutation in the CTR1, or the name suggests constitutive triple response 1, is due to that, because there is a mutation in this kinase which does not phosphorylate. So, there is a continuous activation of ethylene signaling. So, this is the structure of the ethylene receptor ETR1. So, this has the ethylene binding domain, this is a JAB domain which is conserved in evolution, and then histidine kinase domain and a receiver domain. So, some of you may recall I have shown this kind of structure when I was talking about cytokinin signaling. I will just briefly show you about that also. So, this is ETR1, maybe I will go to this part. Now, say this is ETR1, I am showing you these slides. So, this is ETR1 binds to ethylene, change the structure, and if you have a mutation in the ethylene binding domain, then it does not anymore activate, response is off, because it has a mutation in ethylene binding domain. However, mutation in the CTR1 interaction domain is considerably activate, and it is a dominant mutant. So, this ethylene receptor mutant also have been identified in other plants, like in tomato. There is a mutant in tomato which is called never ripe mutant. This never ripe mutant also, like wild type, there is a ethylene response, shortening of the hypocotyl, but never ripe mutant show elongated hypocotyl, as if it cannot read ethylene, and they also do not show ripening. So, even if you keep very long, it does not get that ripe structure, but eventually it will though, but it will never get this color. This is the structure of the ethylene receptor. So, it has, as I was telling you, it is a dimeric protein, it has a ethylene binding domain, transmembrane domain, and a domain in the cytosol which has both histidine and aspartate. So, this histidine gets phosphorylated when ethylene binds, and that phosphorylation can convert to aspartate, and then eventually downstream to other molecule like CTR1, and not CTR1, actually this is a different type of regulation. So, I have shown you these slides when I was talking about cytokinin, because the reason is that This ethylene receptor, cytokinin receptor, both are reminiscents of bacterial receptors. These bacteria have a receptor system called two component kinase system. So, there are two components, one component is called histidine kinase that has a input domain that interacts with the ligand, and there is histidine that gets phosphorylated, and there is a response regulator. Response regulator has a receiver domain that is aspartic acid. So, phosphorylation is called relay of phosphorylation, where phosphate from histidine transferred to aspartate. And also I have showed you this, also I showed you earlier that this ATP is the first time phosphorylation, and then the same phosphate can transfer to the aspartate, unlike the mammalian kinase cascade, where every time one ATP is used. So, this is what in brief how two component system carries on. Now, I will also tell you in brief the differences between actually in plant what you have is called hybrid kinase. Like in bacteria you have seen typical bacterial histidine kinase. So, there are one is the histidine kinase, then you had a aspartate kinase. In plant you have a hybrid kinase where you have histidine molecule and aspartate molecule containing domains in the same protein. So, they are called hybrid kinase. Now, if you briefly see the differences here between the cytokinin signaling, in case of cytokinin, this reception is taking place in the cytosol, where cytokinin is activating histidine kinase and then phosphorylating aspartate, and to carry the signal to the nucleus they have something which is called histidine containing phosphotransfer protein or HPTs. So, there are very small proteins that carry phosphorylated histidine inside the nucleus, and that the response regulators are activated in the nucleus. But this is in contrast in the ethylene signaling, though it begins like cytokinin where histidine phosphorylated aspartate, but the transfer process is different. Here the conformation change of this receptor itself activates the CTR1 kinase. So, this is not a phosphorylay event which takes place in case of cytokinin signaling. So, this is what one student should know, that even though it begins like a same receptor family type of protein, but the signaling component downstream to reception is different. So, this is what actually is like ETR1, this is ETR1 do not response, CTR1 response, and if you have a double mutant then actually it shows like CTR1. So, this is also one student would probably know that this is also called as epistatic interaction, when you make two mutants where you have a contrasting phenotype. When you make a double mutant, then the phenotype which resembles in one of the mutant actually is believed to be working in the down. So, ethylene, ETR1 and then CTR1. So, the double mutant phenotype would behave like the component which is downstream in the signaling pathway. So, that also genetically proven here in this double mutant experiment. So, this is again in the brief about it. So, ethylene, ERS, ETR2, IN4, ERS2, they are all part of the receptor complexes, then the next level kinases, and then comes to the transcription factors. So, this is the mutant I had shown you earlier, CTR1. In CTR1 there is a high level accumulation of ethylene. So, the plant do not grow because ethylene disturbs its growth pattern. These receptors directly interact with CTR1 and affects its activity. So, what I showed you in the diagram, but you may wonder how really people know that this protein interacts with this protein. How did the really scientists came to know about it? There are many experiments. I am showing you one experiment which is called as yeast 2 hybrid assay, where in yeast cells if you express these two proteins separately, if they then some selection methods are there where only when they are interacting they would grow or they will make some color. So, with that experiments they would know that two proteins are interacting with each other. So, CTR1, now what we do not know is how CTR1 really activates the downstream, which is IN2. IN2 or ethylene insensitive 2 is a very important component in ethylene signaling. Like this is what the diagram shows here. CTR1 is a kinase and is also known that IN2 gets phosphorylated, but exactly it is not known whether it is a direct target or indirect target, and that pathway is still not clear, but there is something very important gene called IN2 which is present. So, maybe I will just a little bit go a little fast here. So, here actually IN2 is subjected to proteolysis in the absence of ethylene. So, what happens is that this is a very important protein where which is required to response ethylene or response on. But if ethylene is not there, then IN2 gets broken down. So, response is not there, but when ethylene is there, ethylene inhibits the breaking down. So, it stabilizes IN2. But quite interestingly, still some more interesting thing happens. Maybe I will show you one of the diagram in the down here. Maybe let me go to this slide here. Okay. See here, this is it IN2. IN2 is a protein where it is really quite interesting. This is very important component. If you make a IN2 mutation, then ethylene signaling is absolutely blocked. It is a positive regulator. This ethylene IN2 is a protein that activates downstream kinases. There are many MAP kinases and activate transcription factors. So, this is inside the nucleus. This line shows nuclear membrane. So, inside the nucleus, transcription factors like IN3, ERF1, they activate by IN2. A recent paper actually showed very interesting thing. Yeah, this is what I was showing in a recent paper. What they showed that in this is IN2 protein that has a domain which is this IN2 is a membrane anchored protein, ER membrane anchored protein. What they show that there is also a cytosolic domain of the IN2 that is called C end or C terminal end of the IN2. In the presence of ethylene, there is a cleavage takes place in the IN2. So, IN2 the main part remains in the membrane and a cytosolic part which was becomes soluble and that go to the nucleus. And what happens actually, this C terminal end now will prevent degradation of transcription factors. So, I will show you with this cartoon which is kind of a summarizing slides for me also. Like here, this is ethylene off, this is the receptor complex I showed you, this ETR1, CTR1, and this is cytosolic, it does not go to the nucleus, and in the nucleus transcription factors are made and rapidly degraded. So, this is showing the degradation of transcription factors. So, transcription is shut off. When ethylene is there, then this opens up, this kinase activity is turned off. When the kinase activity is turned off, then this C terminal end of the IN2 is cleaved, that goes inside and that stabilizes the transcription factors. So, this transcription factor stabilization is also important, which you might have noticed in case of oxygen signaling. The oxygen transcription factors also get stabilized by when oxygen is there and due to the inhibition and the inhibitor degradation. Here is also stabilization of the transcription factor by this factor which allows the transcription turned on, which are downstream to ethylene responses. So, this is a brief about ethylene summary what I told you. This is SAM, S-adenosylmethionine makes ACC to ethylene, two genes ACS, amino carboxy cyclopropane synthetase, and SCC oxidase that makes ethylene. CTR1 is a reached by ETR1 complex. CTR1 IN2 is the regulator, response regulator, and then IN3 like proteins and ERFs are the transcription factors. So this is overall summary. Again I am showing the same thing what I showed you earlier, and this one eventually affect cell elongation, oxygen synthesis, fruit ripening, senescence and pathogen defense. And I thank you very much for your attention. I hope this will be useful for your study. For any query, any question, please do not hesitate to write me. If you need a study material, you please write me, I will send you. If you have any question, please email me. Thank you so much.
Thank you so much, sir. Definitely, students. This lecture was very explanatory. He talked about ethylene, the signaling, and also he spoke about how cytokinin and ethylene signaling is similar in some aspects. Definitely, students, go over this lecture once we upload it onto the YouTube channel, and do send us your queries and questions on the email ID that we will display on the screen. So, thank you, students, and join us again for another science lecture tomorrow. Thank you.
Thank you.